皆さん、この素晴らしいイベントに参加できて大変嬉しい思います。This is actually my first presentation in Japan. I'm from Taipei, Taiwan, and I was just told that the temperature today in Taipei is only 1 degree Celsius higher than that in Tokyo. In Kyoto, sorry. Yeah, Anbogen Therapeutics is a clinical stage biotech company founded in Taiwan, and we have 2 clinical assets. One is a precision anti-HER2 kinase small molecule inhibitor, and the other is also a small molecule, which we are planning for a phase 2 study to combine that with immune checkpoint blockade. All right, ABT-101 is a covalent binder of HER2, and it actually has a preferential inhibitory activity for HER2 versus wild-type EGFR. And right now, ABT-101 is under a phase 1B clinical trial. I'm sorry. Okay, so in terms of the non-small cell lung cancer, there are several kinds of mutation within the HER2 kinase domain. The majority of the mutant type is of the so-called insertion type. There are 4 amino acid YVMA insertion into this site. And there are also a number of other insertion types, as well as a small portion of the point mutation. So right now, under the phase 1B dose escalation trial, we started with 100 milligram BID, and right now there are patients under treatment in the 150 milligram BID regimen. Compound, and also we have this is just to show you that compared to one of the competitors, Paratinib, we learned that Paratinib did not show efficacy for cancers with this particular type, G776 VC insertion type. And we have a patient with this insertion type, actually, from her X-ray chest X-ray. Seems like her tumor is responding to our drug. So that's quite exciting. Regarding ABT-301, we believe it is a drug candidate with a best-in-class synergistic anti-cancer activity when it is combined with the immune checkpoint inhibitor. So the mechanism of this protein is a protein deacetylase inhibitor. As we all know that many proteins, including histone and many PD-L1, HISHA protein 90, etc., the protein function can be regulated by acetylation or deacetylation. And here is the structure of ABT-301. And by saying that this is potentially a best class HDAC or PDAC inhibitor, is that this molecule has already completed a phase 1 clinical trial in human with 23 subjects. In accordance to its pharmacokinetics and the safety profile, it is indeed the best, the most safe, and the best. The patients has the best drug exposure in their plasma, high rate and endurable response. So. In reference to the phase 2 direction, there are actually a number of trials already combine typical HDAC inhibitor with the immune checkpoint inhibitor. So one of them is just recently we heard a report from Merck. So here is our team, and myself. We are lucky to have 3 physicians to join us. And our SAB members include Joe Xu, and he is actually the inventor of Alenta during his tenure at Eli Lilly. And Xunbang Xue is also 3 co-founders, including myself, Joe and Xunbang. Both of them are very thorough medicinal chemists. And we also have Dr. Xinchen Gong and Kashan. So here is our plan for the ABT-101. Once we see the solid...
You have to close quickly.
Once we see the solid clinical efficacy for non-small cell lung cancer, we'd like to initiate a basket trial for other solid tumors harboring HER2 mutation. And as I said, we have 2 things in mind for the phase 2 study for the combo. So here today, I would like to invite maybe future fundraising opportunity here, and also future further for partners to collaborate with us. Thank you.
Thank you very much.